Animal research facilities are built around control. Controlled environments, controlled access, controlled health status. But for many facilities, one of the most critical control points in the entire operation still depends heavily on manual processes, decontamination, and that creates a problem. Because when contamination control relies on backpack spraying, manual wiping, or inconsistent fogging methods, variability enters the process, the exact thing these facilities work so hard to eliminate. If your Sentinel program has ever flagged an unexpected pathogen, or if you've had to interrupt a study due to suspected contamination, you already understand the consequences: lost animals, lost data, timeline delays, and increased institutional pressure. Most of the time, the issue isn't intent, it's inconsistency. Animal research environments require more than routine cleaning. Between cohorts, rooms must be fully decontaminated, not just visually cleaned. That distinction matters. Cleaning removes visible debris. Decontamination reduces microbial burden to a level that protects the next study population. But when the process depends on manual application, coverage becomes limited by what staff can physically reach, how consistently protocols are followed across shifts, and how much time is available between cohort transitions. High-risk areas like cage wash spaces, procedure rooms, barrier transfer zones, airlocks, and transition areas can all become contamination entry points if coverage isn't complete. Many facilities are already familiar with vaporized hydrogen peroxide, or VPHP. But traditional high-concentration systems often come with operational complexity, specialized training, bulky equipment, extensive safety considerations, and difficult in-house management requirements. That's why some facilities moved away from VPHP altogether, or outsourced the process entirely. But not all hydrogen peroxide systems operate the same way. CURIS systems' autonomous biodecontamination systems solve these problems at the process level, not just the surface level. Instead of relying on manual coverage, automated systems distribute hybrid hydrogen peroxide vapor throughout the treatment space, reaching equipment interiors, difficult geometries, and surfaces manual methods routinely miss. And because the cycle is automated and repeatable, every room receives the same validated treatment, regardless of who runs it or when. That consistency matters for SPF programs, barrier facilities, biosafety operations, and any environment where study integrity depends on contamination control. For ALAC-accredited facilities and institutions operating under biosafety protocols, automation provides another major advantage: documentation. Cycle parameters, exposure time, and completion status are automatically logged, creating audit-ready records without relying on handwritten notes or memory after the fact. One of the most common applications for autonomous decontamination is between cohort room turnover. When one study ends and another is scheduled to begin, the timeline for decontamination is often tight. Autonomous systems can run overnight or during downtime without requiring continuous staff presence while still delivering consistent, documented results. That means reduced variability, improved facility readiness, and stronger protection for study integrity. The CURIS 3 was designed specifically for these environments. Portable and self-contained, it deploys directly into the treatment space and runs a defined biodecontamination cycle using hybrid hydrogen peroxide technology. No manual spraying, no inconsistent coverage, no guessing whether critical areas received adequate exposure. For facilities managing rotating cohorts, compliance requirements, and tight turnaround schedules, autonomous decontamination isn't just an upgrade, it's a more reliable way to close a critical contamination control gap. In animal research, consistency protects everything: the animals, the studies, the timelines, and the data. And when contamination control still depends on manual variability, the risk never fully disappears. Autonomous biodecontamination helps facilities standardize what matters most, delivering repeatable, validated results with less operational uncertainty. If your between cohort decontamination process still relies heavily on manual methods, it may be time for a closer look. Talk with CURIS system about your facility's contamination control strategy.